Hello, everybody. My name is Aman Chauhan. I'm an associate professor of medicine, medical oncologist, and I lead our neuroendocrine oncology program at Sylvester Comprehensive Cancer Center at University of Miami. I also co-lead our theranostics drug development program at Sylvester. Today, I would like to take this opportunity to throw some light on this novel combination PRRT study. It's a phase 1 first-in-human study evaluating a very brand new drug called piposertib in combination with standard of care Lutathera in well-differentiated neuroendocrine tumors. And the study is known as ETCTN 10450. So we all know about the success story of Lutathera and PRRT. You've heard about the NETTER-1 study, which led to the FDA approval of Lutathera in the United States. And the reason why we like Lutathera or PRRT for that matter for treatment of neuroendocrine tumor patients is because we are able to effectively see the cancer and then treat it. So this is a tailored or precision way to give radiation to the tumor without or circumventing some of the side effects. This is one of our patients from our clinic who received PRRT or Lutathera. If you see on the left-hand side of the screen, there is a lot of disease, including a lot of bones. There's, in fact, not a single bone left without involvement of tumor, and this patient was in considerable amount of pain. Then underwent 1, 2, 3, and 4 rounds of PRRT, and on the right-hand side, you see the outcome, almost 63% reduction in molecular tumor volume. But more importantly, the patient had a significant improvement in disease control and pain. So this is the success story emblematic of PRRT in the current landscape in neuroendocrine cancer. However, since PRRT is now mainstream, should we stop further drug development? And the answer is resounding no. Why? Because there are still a lot of things that we need to tweak and improve on. So the next generation of PRRT studies are going to look into how we can improve on the current success story of Lutathera. How can we make the benefit last even longer? How can we make Lutathera shrink the disease even more than what it does right now? How can we reduce some of the long-term side effects? And those are some of the key questions which are heavily discussed and debated in neuroendocrine cancer societies between our peers, and we try to come with innovative solutions for those. One such solution is trying to combine a radiation sensitizer to PRRT with hopes that the radiation sensitizer would make PRRT work even better. And this particular study is using one such radiation sensitizer called piposertib. So let's kind of dive into a little bit of the background. So what does radiation do? Radiation, be it external beam radiation or molecular targeted radiation like Lutathera, it really kills cancer by causing breaks in the DNA. However, whenever a living cell, for example, tumor, or for that matter, even normal human cell, has a damaged DNA, the intuitive nature of the cell is to repair that damage. And one of the key protein or enzyme responsible for that healing process for the DNA is the DNA-dependent protein kinases or DNA PKs. This led to an opportunity for targeting cancer cells where, especially when we try to target the cancer cell with radiation, and that was by targeting this repair enzyme. So what if we make a drug that blocks this enzyme, and then we induce the DNA damage, but at the same time prevent the repair? This is a very complementary and synergistic approach. So we try to effectively enhance the activity or anti-cancer activity of the DNA damaging agent. And this is where this particular drug called piposertib comes into play. When I first started working with this drug in the lab, it didn't have a name. We used to call it M3814. And I was really, really excited when I first got my hands on the drug. It was shipped to us from Germany, and we got a grant from NCI to study it in our preclinical models. And what we saw when we studied it in mice model using some of the human pancreatic neuroendocrine tumors, we had 4 different set of mice. One where the tumors were treated with a placebo. That means it wasn't an anti-cancer drug. And as expected, you would not see any effect on tumor growth. So the tumors were the largest there. When we treated the tumors with just the piposertib, the M3814 drug, as expected, you would not see any tumor shrinkage, because for DNA PK inhibitor to work, for our drug to work, you'd have to cause that DNA damage. So we haven't radiated a tumor, so you would not see any improvement or benefit there. The third group of mice got radiation by itself. And we know radiation by itself works really well. And you do see the effect and benefit of that. The tumor growth really halted. However, the best results were seen when we combined the radiation with a DNA repair inhibitor, the DNA PK inhibitor. And there we saw both the maximum growth stunting as well as the more durable response to the combination. Based on that, we designed this study, which was first in human study, testing this novel drug called piposertib in combination with a standard of care Lutathera. And the key goal for the study is to look for what is the safe dose of piposertib that can be combined with Lutathera. And that's why we tested in a clinical setting called phase 1 clinical trials. And I'm happy to announce that we now know that what is that recommended phase 2 dose of the combination. And very soon we will be opening an expansion cohort in the study with that particular dose. So we have treated enough patients to have that safety dose, and we will be disclosing it in one of the future meetings this year. Some of the other objectives in the studies to see whether giving this particular drug has some sort of benefit in shrinking the tumor. So we'll be capturing those data points, the RECIST responses. Those are fancy ways to know how much is tumor shrinking on a CT scan or an MRI scan. And of course, we are also looking at how well these patients are doing overall in terms of disease stabilization, so what we call progression-free survivals. Who are the patients who might benefit from this study? These are patients who have well-differentiated neuroendocrine tumors, especially coming from mid-gut, small bowel, or pancreatic neuroendocrine tumors, who have at least been through one line of therapies. And most commonly, this would be somatostatin analogs like sandostatin or lanreotide. Patients otherwise should be fit and normal organ function. And that's pretty typical for most of the clinical trials as eligibility. This is the general schema of the study. Patient would still get standard of care therapy. I'm a big believer patient at least should get what's considered standard so that none of our patients get inferior treatment. However, on top of that, patient who qualify for the study would also have an opportunity to try these pills for 21 days at home during each cycle of Lutathera. And the goal is that together with Lutathera, the piposertib would help slow the cancer healing process so that we can induce durable cancer stability and even shrinkage of the disease. Besides studying the combination safety and effectiveness of this combination, we also are always looking for opportunities to advance our knowledge about this rare cancer called neuroendocrine tumor. So there are a lot of correlative studies or side studies as a part of this study where we'll have opportunity to study the tumor genomics, study the blood tissue of the patient to kind of figure out, is there any way to find better biomarkers to monitor the disease, find biomarkers that can predict responses to the disease, that can predict side effects to the disease. So this study really helps the field, the community, and the future generation of patients to help advance us knowledge about some of these finer aspects. So there's a lot of science involved in the study. And lastly, I'd like to thank, for any study, no matter how big or small, it literally takes a village. In fact, I would say a county to kind of make it happen. So I'm very grateful to my mentors, collaborators from across the US and Canada, and most importantly, our patients, without whose support we would not be able to bring novel, innovative solutions to our current problems. With that, I would like to thank LACNETS for this opportunity to educate our patients and caregivers about key clinical trials currently ongoing in the US and Canada. And for more details, please feel free to reach out. And you can also find information at ClinicalTrials.Gov. Thank you so much.